So at this last AACR meeting, there was some nice research done. There's been some small incremental advances in cancer research and in treatment of patients. Still, there's not been a big major breakthrough in terms of treatment. The major breakthroughs have come in terms of understanding the genes that are involved in cancer and analyzing the specific tumors from the patient and figuring out where those mutations are that lie within the gene. So you may know that 85% of cancers out there are due to mutations in the genes, whether it's environmental, whether it's diet, whether it's even endogenous. There somehow, some way, some genes get mutated, and those are the genes that drive the cancers. So it's going to be pretty routine in the future for if someone gets a tumor and it's caught at a certain time, and they typically do what's called a biopsy. They will go in and they will take a little piece of it, and usually what they do now is they'll take it and they'll embed it in some parafilm and put it on a microscope slide, and then they will have a pathologist take a look at it, and the pathologist is responsible mainly for grading the tumor and just by the way that it looks, the way that the cells look, and by sort of announcing what the stages are. I think radiologists also get involved. They will take MRIs, or they'll take PET scans or CT scans of the tumors as well if they can, and they help to diagnose the type of cancer that it is and what stage it is. Increasingly, they're starting to take these little biopsies and they're being able to do genetic analysis. And the genetic analysis is very powerful. They can find out really all the mutations that there are in all the genes from that little biopsy. We're talking about very small amounts of tissue. But and the goal is to actually create medicines or prescribe medicines that are very specific for that particular mutation signature that's in the cancer. The problem is that those are not very specific. The kinds of medicines that are out there basically attack, for the most part, they attack rapidly dividing cells. And oftentimes it's very difficult to deliver the drugs to their destination. So I was thinking when I was there, and actually I've been thinking about this for about a year, and I haven't come up with a good solution, so I thought that I would pose the problem to you guys and see if you guys can talk about it with your friends and think of a good idea. You guys are young and your minds are fertile, okay? So one of the things that would be great would be to come up with a drug. Let's say, for example, you have something called a messenger RNA. I know we haven't talked about this. You will get this in Chem 431C. Okay, there's messenger RNA. And the messenger RNA is actually made from the DNA. So DNA is double-stranded. So I'll just kind of draw it as a ladder here. Okay, and it makes this messenger RNA. And if the DNA has a mutation, let's see if I can do this here. So if it has a mutation somewhere there, that mutation will be transcribed, so now you'll have a base change, okay? And let's say the base change is from A to C, okay? So now you have a single base change there. It would be great if someone could design a drug that would be composed of the following. The drug would be basically something that would be maybe two different parts, okay, and it would bind specifically to that particular mutation and maybe a couple of base pairs or bases upstream of that. This is the 5', this is the 3'. And then you have this other part here that would bind just down there. So it's like a two-part drug, okay? And then if it had a toxin, okay, so that's a toxin there. And then what would happen is that as the DNA, as this hybridization comes together and gets very close to this, that this would get released, maybe by hydrolysis. You guys know a lot about hydrolysis now. By hydrolysis. And this then becomes active, and this will kill the cell. Okay? And things like this have been thought of, but I haven't really gone into detail. Nothing was talked about at the ACR meeting, but they've been thought of by other people that I know in the cancer field, people that have been long thinking about cancer. And in this stage, it would be called the prodrug. Okay, why is it called the prodrug? It's called the prodrug because it's not yet toxic. Okay, because when it's bound to here, it's not going to be toxic. But as soon as it's released, now this is the active drug, and now this will kill the cell. Now, there's a lot of limitations of this idea, but this would be a major breakthrough if someone could think of a way to design such a drug. I have it written as a two-part drug. Maybe it doesn't have to be a two-part drug. Maybe it could be a one-part drug. But something that would be very, very specific for cancer cells. Now, why is it specific for cancer cells? Because only cancer cells will have that mutation there. Okay, and it has to be small enough to get through the plasma membrane and small enough to get all the way to the messenger RNA. But if enough of this could be made, you can get a lot of specificity due to the messenger RNA, and then you could hopefully kill the cell. So hopefully one day, I mean, this is just one of many ideas out there. Hopefully one day we can be more specific in terms of cell killing so that we do not kill normal cells, but we kill cancer cells. So that's my little two-bit idea about how we can go forward with cancer and coming up with effective medicines that are specific. Because right now it's rapidly becoming the most common killer of people in the United States. Okay, today I want to talk about a little bit more about this chemiosmotic theory. This was a theory, this is the same thing as PMF. Okay? This is the theory that was put forth many, many years ago in the 1960s, maybe 1950s, by a person by the name of Peter Mitchell. Okay, so here's Peter Mitchell. Okay, and at the time, this was thought to be a very crazy idea that oxidative phosphorylation could occur by a proton motive force. I'm going to, actually I have this diagram up here. You'll get this diagram also when I put this up on Moodle. But let me just go ahead and sort of explain to you experimental proof that his idea, called the chemiosmotic theory, has turned out to be true with experimental proof. So it turns out that there are these bacteria called purple bacteria that live in the ocean, and they have on their membrane surfaces, on their membranes, a protein known as bacteriorhodopsin. Okay. And bacteriorhodopsin is a very interesting molecule. It's a transmembrane protein. Okay. It's purple. It gives this purple color because it has a dye in there. I shouldn't say a dye. It has a chromophore, okay, which is a small molecule that absorbs light. It absorbs sunlight, and they look purple when you look at them, actually even in the ocean, but also under the microscope. And what happens with these bacteria is that they can actually pump protons across this membrane due to this bacteriorhodopsin. This bacteriorhodopsin uses sunlight in order to capture protons and put them inside the bacterium. So, and they use this actually to create ATP for themselves. So they thought, okay, let's go ahead and use this bacterial rhodopsin and see if we can really prove that the chemiosmotic theory is correct. And so what they did, experimenters did, is that they took an artificial vesicle called a liposome. Okay? And a liposome is basically a membrane, okay, that's formed of these diacylglycerides, okay, and so that's the membrane there, okay, and they were actually able to embed this bacterial rhodopsin into this artificial membrane. So that's the liposome, okay, and this is the bacterial rhodopsin right here. And sure enough, when they did shine light, we'll just sort of make this like the sun. Okay, so here's the sun. Okay, when they shine light onto this system, they were actually able to get protons to go into this liposome. Okay. Then what they did is that they said, okay, we're going to actually create ATP. So we're going to put in the bacterial rhodopsin, which I'll just abbreviate as BR, and we're also going to put in the ATP synthase, okay. And when they did this, and they added ADP and inorganic phosphate, they were actually able to create ATP in the presence of sunlight. Okay, so you need sunlight or any light that shines white light on this. Okay, so they're actually able to create ATP with this system. One of the important controls that they did for this, okay, so here's a control experiment. So when you're an experimenter, you always got to think about controls, and positive controls and negative controls. So I guess this is a negative control, okay? And so the negative control, okay, is to put in BR, okay? And of course they've got this ATP synthase thing going on, okay? And then they add in dinitrophenol, okay? What do you guys think is going to happen when they put in dinitrophenol and they put in ADP plus inorganic phosphate going to ATP? What do you think is going to happen when they put in the dinitrophenol? That dinitrophenol should ring a bell to you. Okay, it will inhibit. Very good. So you will get no longer any ATP synthesis. Okay. Okay. Now the question is, how does this dinitrophenol? How does the dinitrophenol do this? Very good, very good. It acts with the protons. So we have a particular name for this type of inhibitor. Does anybody remember the name for this type of inhibitor? You're right, it will react with the protons. It starts with a U. Uncoupler. Uncoupler, all right. So this is an uncoupler. Okay, very good. And so what happens is that even though this is actually producing, you're actually making protons go across here with the sunlight, the nitrophenol actually brings back the protons back across the membrane. So it dissipates the PMF. There's no proton motive force, so there's no ATP synthesis. Okay? So it's a beautiful way of showing that Peter Mitchell was right, and a number of other biochemists were actually wrong because they thought of other ways in which oxidative phosphorylation occurs. But this is actually really strong proof that you need to have the proton motive force built up in order to create this ATP. So it's a very simple system, but very effective. Okay, I wanted to also talk about another function of the mitochondria, and that is apoptosis. Okay? So in order for me to talk about this, I have to talk a little bit about what apoptosis is. Apoptosis, anybody know what apoptosis is? Anyone heard about this? What is it? Cell death, yes. Everyone, yes. Cell death. I call it programmed cell death. Okay? Because there's a definite series of reactions that take place in order for the cell death to occur. So you can kill cells by throwing bleach on them. That's one way. Okay, we call that necrosis. Okay, but this is a different type of cell death. It's a cell death which is pre-programmed, ready to go in case something goes wrong. So this is not necrosis. So this is actually apoptosis. Sometimes it's pronounced apoptosis without the second P being pronounced. Okay, so apoptosis or apoptosis, either one is fine. Well, apoptosis is really, really important. We've known about it for about maybe 10 years now, maybe 15 years. It's been popular, and when does this actually take place? Well, I can think of. Let's see if I got my notes here. There's actually two types of classes of apoptosis, which I'll divide into developmental and what I'll call defensive. Okay. Developmental means as the organism is going through morphological changes, as it's being built up to be born, okay, we call that developmental apoptosis. And for example, I'll give you 3 examples of developmental apoptosis. Number 1, cells between digits will die. Okay. When I say digits, I mean, for example, fingers and toes. Okay, so there's a case where if you look at photos of developing embryos, you'll notice that there's webbing between the fingers and the toes. And obviously when you're born, you don't have that webbing any longer, and so the cells will actually die. So there's actually apoptosis causes that cell death to occur, so that now there's no more webbing between the digits. There's actually a syndrome which is called syndactyly. Just write that for you. This is a case up here where you have syndactyly. Where there's insufficient apoptosis occurring between these 2 digits here. And so that's why you can see that they separate very high up there on the toes. Okay? So cells between the digits die. Okay? Another case of apoptosis, developmental apoptosis, is the tail of a polywog. Tail of polywog. And you guys know that polywogs eventually become frogs, and frogs don't have tails. But in the beginning, you do have these tails of the polywog, and they die through a process known as apoptosis. third case of developmental apoptosis is our immune cells that travel through the thymus. Okay, so humans, when their immune system starts to develop, that immune system can attack not only foreign epitopes, okay, but they can also attack the self as well, okay? And so there has to be a way of getting rid of those immune cells, those T cells that attack the self. And you probably know that there are some fairly devastating disease like lupus where that ability to attack the self somehow comes back. It's not clear how, but somehow it comes back. Well, it turns out that, again, during the fetal and embryo development, I guess fetal development, there is a mass migration of what are known as T cells. You guys may have heard of T cells that are part of the immune system that go through what's known as the thymus. And when they go through the thymus, they actually start to go through apoptosis if they recognize what are known as self-antigens. So in other words, epitopes that you would see on your own cells. Okay? And that's actually why the T cells are called T cells, is because at one point they had to have migrated through the thymus in order to become educated, that's the word educated, in order to not react with the self. So if that doesn't happen, then you have a lot of cells that will cause what are known as autoimmune disease. And those autoimmune diseases can devastate the person. There's a whole series of these autoimmune diseases where maybe a few of these T cells somehow escape this education process by going through the thymus and becoming annihilated, okay, or going through apoptosis. Okay, so those are three cases of developmental apoptosis, but as I said, there's another type of apoptosis known as defensive apoptosis. Okay. And defensive apoptosis occurs in adults. Okay. And what it is that if you are you want to skip school one day, it's a nice bright sunny day, you head on out to Santa Monica, you put your towel out there on the beach, and you've got your iPhone plugged into your ear so you can listen to some great music, and the sun is beating down on you. Well, a lot of your cells are going to be going through apoptosis, just to let you know, okay? And why is this happening? Well, it's happening because the sun has ultraviolet light, and that ultraviolet light causes DNA damage in your skin cells. And in order for you not to get cancer, okay, those skin cells will die, and they die through a process known as apoptosis. So it's really important that you have apoptosis. That's not the only case where you have to have defensive apoptosis. Now, why do I call that defensive? I call it defensive because if you don't go through apoptosis, if some of these cells escape programmed cell death, but yet they've gotten DNA damage, then what happens is those cells could divide uncontrollably, and you can get skin cancer. And as a matter of fact, there's a whole education process that people need to go through in terms of going to these tanning salons, right? These tanning salons where people think it's really healthy to have kind of like brown skin, but what happens is that those can be very, very dangerous. So you got to be careful about protecting your skin when going out there. Apoptosis also occurs in your colon as well. So when your colon meets up with a toxin that's found in some food, and that toxin causes some kind of mutation, then your colon cells know to commit suicide rather than risk the possibility that you could cause cancer for the person who's just eaten whatever it is. Okay, so you've got all kinds of defensive apoptosis, and I forget what the rate is, but there's a lot of cells per day that are dying because because of DNA damage causing apoptosis. So I'll just give you one of these examples of defensive apoptosis. And that example is going to be related to mitochondria. So I'm going to relate this to mitochondria. Okay, so what happens is that in defensive apoptosis, which can occur due to UV light exposure, it can also due to heat, excessive heat. It can be due to something called ionizing radiation. Like, for example, cosmic rays or X-rays. Those are examples of ionizing radiation. And also chemicals. Okay, just give a thing called chemicals. Okay. And what happens is that in the mitochondria, you've got this outer membrane, and you have this inner membrane, and there sits on this inner membrane the whole electron transport chain, but I'm just going to concentrate on this one, which is called cytochrome C. So cytochrome C, as you know, is a carrier of electrons. And the electrons eventually go to complex 4, and then complex 4 then will reduce oxygen into water. Well, it turns out that in apoptosis, defensive apoptosis, a pore, well, that didn't work out well. Okay, a pore will open up in the outer membrane, and this cytochrome C will come out, and it causes activation of an enzyme known as caspase. And caspase is actually a protease, okay, similar to trypsin, I guess, protease. And this actually starts the apoptosis program. That actually initiates the apoptosis. And so all it takes is for enough of the cytochrome C molecules to come out and activate caspase, and that will cause apoptosis. Well, how does this happen? How does it that the cytochrome C knows to come out? How does it figure out that now is the time to make the cell go through apoptosis? Well, we have to bring in a molecule that I work on quite a bit, and it's called P53. And P53 is this tumor suppressor protein. Okay. And what happens is that P53 recognizes DNA damage. Okay. And I won't go through exactly how. I could do that offline, another time. But basically, it recognizes DNA damage, and DNA damage will basically activate this P53 tumor suppressor protein. Okay. So P53 when it's active, binds to DNA. So I'll just sort of right here, here's P53, here's DNA. Okay. And what it does is it causes the increase in the level of messenger RNAs of certain genes. Okay. And one of the genes that it increases the messenger RNA level of is called BAX. And BAX is a very important gene because the messenger RNA will go ahead and make the protein, BAX protein. And that BAX protein has an interesting domain structure. So if this is the amino terminus end of the protein, and this is the carboxy terminus of the protein, okay, it has what are known as domains. So here's BH3, BH1, BH2, and something called TM. Okay. So those are different regions of the protein. Okay. So of course we start off here with methionine, and I don't know what the carboxy terminus is, but and so these are different regions of the BAX protein. And this is required for what's known as oligomerization. Okay, so that causes BAX proteins to come together, other BAX proteins to come together, almost like glue. This is important for pore formation. This is also, BH2 is also important for pore formation. And this is called the transmembrane domain, okay, the TM. So what happens is that this BAX protein, once it's made in the cell by p53, which has just been activated by DNA damage, it will cause a pore to open up in the outer membrane of the mitochondria. Okay? And the name of this pore, and that happens because this protein really starts to bore in here, and it starts to make this hole in this outer membrane. And the name of this pore is called the mitochondrial apoptosis inducing channel, or MAC. Wait a second. MAC, mitochondrial apoptosis inducing channel. Okay. And once this cytochrome, that allows the cytochrome C to leave. Okay, so it's going to leave. Okay. And cytochrome C will go ahead and activate, as I said, caspase 9, specifically 9, and that initiates apoptosis. Why bring this up? I'm bringing this up because I wanted to show you that a lot of times these proteins have many different functions. They are used for multiple pathways inside the cell. So while cytochrome C, I told you, is essential for electron transport chain, yes?
Is that caspase 9, the Cas9 that's not being used in CRISPR?
No, that's a really good question. So the question is, there is a new technology called CRISPR-Cas9. I think it's CRISPR-Cas9, okay, which is used for something called DNA editing. Okay, and it's been taking the popular press by storm. People think you can make designer babies out of these, and I've looked at the technology. It's a lot harder than one thinks, and there's a lot of off-target effects. So I think in the laboratory it's a great tool, but I don't think we're going to see any designer babies coming out of CRISPR-Cas9 in the near future. So this is not the same as caspase 9. Caspase 9 is a protease, right? Okay, maybe you guys can talk. We can talk about CRISPR-Cas9 in CHEM oh gosh, I don't even know what the name of it is. Anyway, whatever the next semester is in the chemistry series. Okay, so but the reason why I want to talk about this is I want to show you that cytochrome C has other roles besides just electron transport. We are ready now to enter into a new chapter in the book, and the chapter is about photosynthesis. Okay. The overall reaction for photosynthesis is CO2 plus H2O. It's going to make O2 plus carbohydrate, which I'll just abbreviate in general as this. Okay, so that's carbohydrate. Okay. And in plants, photosynthesis takes place in organelles known as chloroplasts. Chloroplasts are a lot bigger than mitochondria, okay? But they do have an inner and outer membrane. Inner membrane. Has an outer membrane. They also have what are known as internal membrane structures. And those are known as thylakoids. And inside the thylakoids, we call that a lumen. Okay, lumen. Okay. And there's something called grana. There's a lot of these terms here. Grana. And those are stacks of thylakoids. And there's this other term called lamellae. And lamellae are thylakoid membranes. So a lot of new terminology here. Okay. And finally, there's something called the stroma. And the stroma, this is outside of thylakoids. But inside the chloroplasts. Okay. In chloroplasts, and this is essential to remember, in chloroplasts, the protons are pumped actually the proper term is translocated into the lumen of the thylakoids. Okay? And we're going to find that pumping is going to be very similar to the translocation that we found in mitochondria. And it's going to be important for driving ATP synthesis. Let me go to your book and point out some of these structures here. have this nice overview, which I return to because I think it's a nice organizing principle. So we've talked about polysaccharides and monosaccharides. We've talked about the glycolysis here. We've talked about all the way down to electron transport and ATP synthesis. Okay. And we've talked a lot about gluconeogenesis as well. So that's going to be an anabolic pathway. Okay. And then here we have on this right-hand side this sort of photosynthesis taking place. And let's see if this really makes sense. So in photosynthesis, we've got the oxidation of water to oxygen. Okay. We have the take-in of CO2. So CO2 actually is fed into the photosynthetic machinery. Okay. And that CO2 is going to be eventually used to make components of these carbohydrates. Basically, it makes glyceraldehyde 3-phosphate. You've heard of that term. And glyceraldehyde 3-phosphate will make glucose, and you can store. And then the plant stores these polysaccharides. Okay. So I'm trying to think here. I guess the carbons come from the CO2. So even, this is like the opposite of catabolism. This is anabolism. So this is we're actually using the carbon dioxide in the plant to make glyceraldehyde 3-phosphate. That is going to be a reductive process. In other words, we're going to have to reduce the CO2 in order to make the glyceraldehyde 3-phosphate. We're also going to need ATP in order for this process to occur. It's an anabolic pathway, so we're going to need ATP. So we need two things to happen here. We have to get some kind of reduction going on, and also we're going to need ATP. It's going to require some energy, some chemical energy. Where do we get the energy from specifically? I guess what's going to happen here is that this the protons are going to come from the H2O. Okay, the protons will be stripped from the H2O, and that's why water is reduced compared to the O2. That makes sense. We're going to get our protons from here. Okay. And we're also going to get electrons out of this as well. So we're going to get electrons out of the H2O as well. The electrons are not going to be used for the protons. Protons have no electrons, but the electrons will be used to eventually reduce the CO2. What is the form of the electrons that will be used? They're going to be used in something very similar to NADH, but instead of NADH, it's called NADPH. NADPH is where we get our reduction potential from. Okay, so in other words, these electrons in this water will reduce NADP plus to NADPH, and NADPH is used to provide the reduction of the CO2 to the glyceraldehyde 3-phosphate. Beautiful system. Pretty impressive. Okay, all this is happening. And of course, ultimately, we have to get energy to do this, and that energy comes from the sun. And out of all the sun's rays that strike the Earth, only 1% of it is actually captured by these plants. 99% is kind of wasted, just out there. Okay? I guess we're trying to capture this in the Mojave Desert with all these solar panels, right? Or on top of roofs. But a lot of the energy is just sort of wasted as far as we know. Okay, so that is sort of an overview here. Let's see if we can make sense of some of these other diagrams in the beginning of this. Okay, so we've got this little diagram here where we've got photosynthesis, and photosynthesis is where you've got the 6 CO2 plus 6 H2Os come together. I guess that's supposed to be glucose or something close to glucose, some kind of sugar molecule plus O2. Okay, so, And you guys now know that that energy is used to strip protons as well as electrons from this water, and that's going to be used to help to reduce the CO2 into sugar. What else is happening? We're also able to use this sugar in order to make more ATP later on. That's respiration. But let's just concentrate on this left part here, this left half. Okay. Anything else I want to talk about? Oh yeah, I guess let's talk about this other diagram here. I wanted to mention this diagram. So here you have something called the light reactions and the dark reactions. Okay. So in the light reactions, oh good, this is actually kind of what I was talking about. Here you have H2O, and the H2O is going to help, well, what's going to happen is that the H2O is going to get oxidized to O2, so let's just go with that, okay? And we're going to use the protons from this H2O to create a proton motive force, and that proton motive force will drive ADP plus inorganic phosphate to make ATP. I think without that good knowledge of the proton motive force, this would kind of not make sense, right? But now that we know about proton motive force, you say, okay, I can understand how these protons here from this H2O can be used to make ATP from ADP and inorganic phosphate. Of course, you need ATP synthase for that. Also, what's going to happen is that the electrons that are stripped from this H2O will be used to convert NADP plus into NADH. And that's going to be through something called hydride transfer. Hydride transfer. Here we're transferring the hydride onto the NADP plus. Okay, that's all. And why they're called light reactions? Because this is taking place in the presence of the sun. Okay, you have to have sunlight or some kind of light for this to happen. Then there are these other reactions that are taking place where basically you have anabolism. You can count all of this down here as anabolism. Here you've got CO2 plus some water, and you're going to require NADH. That's where you're going to get your reduction from, NADH. You're going to require ATP because it's an anabolic process. It requires ATP, okay? And you're going to make your carbohydrates. And us humans, we enjoy this process because without this, we would not be able to get carbohydrates from plants. The plants would not be able to feed the animals that at least some of us eat, okay? And so we wouldn't be able to have animals. And also, another byproduct of this is O2, which we need in order to undergo respiration and electron transport chain. So you can see how it all fits together. Okay. Oh, good. Let's just go through this anatomy. This kind of talks about all those terms that I just explained. So this is on page 676 of your book. Let's just look at this sort of sort of artist rendering of this. I had talked about chloroplast. So this whole thing is the chloroplast. We talked about the outer membrane and the inner membrane of the chloroplast, but that's not a big deal, as much of a big deal as it was in the mitochondria. Okay, where all the action takes place is going to be across this lumen. So this lumen, let's just sort of zero in on that. Uh-oh, I'm not sure what I did. Okay, shoot. Okay, darn it. I hit something. Okay, well that. Come on. Oh, okay. So, gotta make sure I not press so many buttons here. Okay, so I'm trying to point out that there's this lumen inside here, which is where protons are going to build up inside here, and the stroma is the area outside of the lumen. Okay, so this thylakoid membrane is really, really, really important. The thylakoid membrane. You can almost think of it as similar to the inner membrane of the mitochondria, but everything's reversed. Remember, in the mitochondria, the protons are being pumped out of the inner membrane. In the thylakoid, everything is being pumped in to the lumen. Okay, so the protons get pumped into the lumen. And then you have this grana, which is basically stacks of these thylakoids, and the stroma, which is the material which bathes the thylakoids. So remember that word thylakoid, and the lumen, and the stroma. Those are three important things to really remember. And they're green, obviously, because they have chlorophyll, which captures the light energy. And we'll talk about chlorophyll and how this capturing of the light energy is used to excite electrons. And those electrons go through an electron transport chain. And that electron transport chain is what drives the proton motive force, as well as helps to, it will be used to reduce the NADP plus to NADH. So we'll talk about electron transport chain in the thylakoid membranes next time.